Let us take the process of oogenesis now. Oogenesis means egg formation, or the process of egg formation. Oogenesis is a discontinuous process, especially when we are talking about human females. It is a discontinuous process. Once we understand the process, we will understand the meaning of this discontinuous word. It starts in the fetal stage. The process starts in fetal stage and gets completed after puberty, and completed after puberty. And here also there is one condition. The condition is if fertilization takes place. If fertilization takes place. So it starts when the fetus is still in the mother's womb. Then it will get again restarted at puberty and will get completed only if fertilization takes place. If no fertilization, then it is left incomplete. Human females are born, when they are born, the fetus or the female baby is born, it is born with a fixed number of follicles in its ovaries. So now let us see what all changes take place in the follicles. In case of spermatogenesis, we said that germinal epithelium, it undergoes mitotic divisions to form many spermatogonia or sperm mother cells. All this already happens in the fetal stage, and after birth that number never increases. So how many eggs a female would produce in her reproductive phase is decided at the time of birth itself, because all those cells which will give rise to the egg, they are already formed there. So no new such cells will be formed after that. When the fetus is, or let us talk about the gestation period, fetus is in first or after first month of gestation period. There are about 1700 follicles, 1700 follicles. What are these follicles? These follicles are the cells of the germinal epithelium which will give rise to the egg. When the fetus turns 5 months, or let us take one more stage, 2nd month, the number of follicles increases. That means there is multiplication which is taking place. So this number comes to 600,000. 5th month, by the end of 5th month, this number comes to 7 million. At birth, when this baby, female baby is born or baby girl is born, it has a fixed number, and that number is 2 million. So now you can see what is happening here in these numbers. It is only 1700, then it is 600,000, then it becomes 7 million. So up till now, that is up to the 5th month, the number is increasing. So that means multiplication is taking place. At birth, the number has decreased. This is due to a process called atresia. So what happens here is, Atresia is a term which is given to shrinking of a structure, maybe a cell, maybe tissue or an organ. So, here these cells out from 7 million, they are only 2 million which remain, and why this number has gone down? Because some follicles, they undergo atresia, which is also known as atresia, follicular atresia. So, at birth there are 2 million follicles. And when we made the structure of ovary in the female reproductive system, we said there is a cluster of 500 follicles, and only one follicle out of those 500 gives rise to the egg. So, there are 2 million, every cluster of 500, only one out of that 500 will change into the egg. So, the number is decided. Now, at puberty, this is at the time of birth, 2 million. At puberty, the number has or number goes further down. That means atresia continues. At puberty, they have 60,000 to 80,000 follicles only. That means from 2 million, further follicular atresia continues, and at puberty there are only 60,000 to 80,000 follicles which remain in the ovary. After this, multiplication is not taking place. That means the number of follicles for egg formation is not going to increase. So, all these things are in the fetal stage only. Now, we were talking about this multiplication thing. Let us take the stages of oogenesis. Stages of oogenesis. It is completed in 3 phases: multiplication phase, then growth phase, and maturation phase. Multiplication phase is mitotic division, growth phase is growth. Here the size will increase due to yolk accumulation, that is vitellogenesis. We will talk about that. Maturation is meiotic division, where haploid eggs will be formed. In case of spermatogenesis, we saw that multiplication, growth phase, multiplication, growth and maturation, everything takes place in the seminiferous tubules. In case of oogenesis, it starts in the ovary but will get completed in the fallopian tube. We will see how and why that happens. So, now let us take up the various stages of oogenesis in detail. These are the follicular cells. In the fetal stage, they undergo multiplication. So, these have divided, and we get, because the division is mitotic division, all these cells which are produced, they are called egg mother cells or oogonia. Oogonia or egg mother cells. They are diploid, 2n, these follicles are 2n. What is happening here is mitotic division. So, here what is happening is multiplication. So, first phase is multiplication. Where is it happening? It is happening in the fetal life. So, when the fetus is still developing, at that time this multiplication is taking place. Then these oogonia enter the next phase, that is growth phase. So now this, whatever is going to happen here is growth phase. Let us write down what happens in growth phase, because that is very, very important. In growth phase, the size increases. Size increases. This increase in size is directly dependent on how much yolk is formed or synthesized. Just to have some idea about how many times would this increase in size, it is 200 times in human beings, in human females. About 90,000 times in Drosophila. 2,000 times in frogs and 200 times in hen. So there is a variation. It can increase 200 times, 90,000 times as compared to the oogonium. So from the mother cell to the next stage, there is increase in size. Second change that we see in this growth phase, why is this increase in size taking place? Due to yolk accumulation. So vitellogenesis takes place. Vitellogenesis means the process of yolk formation, and for this there is a structure which is called yolk nucleus is formed. So yolk nucleus is formed. Yolk nucleus is actually a cluster of mitochondria and Golgi body which help in vitellogenesis. So because of this yolk formation, the size is increasing. Third change that we see here is growth is not only because of yolk. Now the cell has to divide to produce the egg. So all those proteins, RNA, everything whatever is required has to take place. So protein, RNA synthesis increases. That also helps in this increase, or that also results in increase in size. In other animals, not human beings, other animals means reptiles, birds, the yolk content has to be more, and that is helped by some special types of chromosomes. So these in birds and reptiles, those special types of chromosomes which are known as lamp brush chromosomes are formed. These lamp brush chromosomes, they help in yolk synthesis. In human beings, the yolk content is negligible because throughout the fetal stage, the nourishment is supplied by the mother through placenta, and that is why that yolk, which is the reserve food material, is not required. But in case of birds and reptiles which are oviparous, that means they lay eggs. After they have laid the egg, there is no connection between the female animal and the egg. So if the egg requires nourishment, the female animal or the mother of that will not be able to provide that nourishment. So the egg has to be self-sufficient in that yolk content. So these changes. One more change which is seen, fifth point, at this time a membrane is secreted around the egg. The membrane is known as vitelline membrane. Vitelline membrane is secreted around the egg. The egg itself secretes this membrane, it is called vitelline membrane. So, a plasma membrane and these vitelline membranes, they are almost close to each other. That additional membrane is secreted. Because RNA synthesis and all protein synthesis has to increase, our RNA production has to go up. It is by a process called gene amplification. Gene amplification for our RNA synthesis takes place, for our RNA synthesis. So, size is increasing, nuclear division is not taking place, so chromosome number is same, and we show a big cell which is still diploid. This is diploid. It is known as primary oocyte. Now, the important thing which we need to understand here, multiplication phase takes place in the fetal life itself. When the female fetus is growing in its mother's womb, at that time this multiplication has already taken place. These cells, after multiplication, they enter into growth phase. That means growth has started. After growth, initial growth phase can be divided into two parts. There is an initial growth which takes place and then the later growth which is followed. So, after initial growth, it enters into the maturation phase. Initial growth is slow and less. After this, the cell will enter maturation phase. Maturation phase is meiosis. After it enters maturation phase, that means it will undergo meiosis 1 and then meiosis 2. It enters the phase and prophase 1 just begins and stops. After that, further growth will take place. And after that growth, whatever changes are required, that will start at puberty. So, for how much duration is growth taking place? In case of human beings, from birth, because all this has happened till birth, from birth till puberty. So, the complete period of puberty, that is 11 to 13 years, is the period where growth is taking place. But when does it take place? After initial growth, that is little bit of growth, it enters maturation phase. When it enters maturation phase, it goes into meiosis 1, prophase 1 of meiosis, and the division stops. That means maturation starts and then stops. After that, growth continues. When will this meiosis 1, prophase 1 get restarted? It will get restarted at puberty. So, that is why growth period is very, very long in case of human females. So, now this primary oocyte enters maturation phase. It enters maturation phase. So, let me write this as maturation phase. Maturation phase is meiosis. This large primary oocyte will undergo meiosis 1. Meiosis 1 is reductional division. So, chromosome number has to reduce to half. So, this cell will get half the number of chromosome and the other cell will also get half the number of chromosome. In a normal division, the nuclear division is half and half. If in case of human beings we are talking of 23 chromosomes to one cell, 23 chromosomes to the other cell. And then cytoplasm also gets divided almost 50-50. In this case, nuclear division is proper 50-50, but cytoplasmic division is unequal. One cell gets less cytoplasm and the other cell retains almost complete cytoplasm. That means there are two unequal sized cells which are formed. Nuclear division has to be proper 50-50 so that haploidy can be maintained, 23 chromosomes here, 23 chromosomes here. If the cytoplasmic division was also equal, if we are saying, then this cell would have received half of the cytoplasm and organelle, and the other cell would also have received half cytoplasm and organelle. And we know that only one egg is to be formed. So why waste that half of the cytoplasm? So this unequal division of cytoplasm is to conserve all the cytoplasm and organelle with only one cell, which will give rise to the egg. So this smaller cell which is formed is called the first polar body. There is one more term which is given to these polar bodies, they are known as polocytes. So it is the first one which is formed, it is called first polar body. And the other large cell which has haploid nucleus and has retained the complete, almost complete cytoplasm, it is called the secondary oocyte. We have talked about secondary oocyte. When we said ovulation takes place, the egg which is released is actually in the secondary oocyte stage. So when we say egg is released from the ovary, it is not the egg which is released, it is this stage which is released after meiosis 1. Where is all this happening, multiplication and growth? In the ovary. Up till now, up till secondary oocyte stage, it is in the ovary. After this, the Graafian follicle will rupture and this secondary oocyte will be released. It will be taken by the fallopian tube and here it will wait for some time for fertilization. If fertilization takes place, then meiosis 2 will take place. If fertilization does not take place, then it gets lost from the female body in the secondary oocyte stage itself. But because we have to talk about the entire process, we will complete it. So up till secondary oocyte in ovary, after that in fallopian tube. So now this secondary oocyte undergoes meiosis 2. Meiosis 2 is equational division. That means chromosome number will remain same as the parent cell here. So here also the two cells which are formed, they will have same number of chromosome, that is haploid cell. And again, because only one egg is to be formed, one cell will retain the complete, almost complete cytoplasm, and the other cell will get negligible, whatever is required for the basic division. This again a smaller cell which is formed, it is called second polar body. Actually both the cells should undergo meiosis 2, but in human beings first polar body does not undergo meiosis 2 because anyways this has to degenerate, and if it has to divide, for movement of those chromosomes from the equatorial plane to the poles, ATP consumption takes place. So why waste that ATP when anyways these cells are not going to help, only this cell is required. This cell which is formed is the ootid or the egg or ovum. So haploid cell. Now the things which we have to remember in oogenesis, multiplication takes place in the fetal life of the female, when the female is still in the fetal stage in the mother's womb, multiplication has taken place. After multiplication, these oogonia or the egg mother cells, they enter the next phase, that is growth phase. It enters the growth phase, little bit of growth takes place, Most of the growth is later on. After that, it enters the maturation phase. In maturation phase, it enters prophase 1 and then maturation phase stops. Then the later part of the growth will take place. More and more yolk accumulation, more and more protein and RNA synthesis, all those changes will take place. So, meiosis 1 gets arrested at prophase 1. It will restart at puberty. So, till that time only growth is taking place. So, in case of human females, the growth period is very, very long. It is about 11 to 13 years from birth till puberty. This large cell which is formed after growth is called primary oocyte. It is diploid. It undergoes maturation phase, that is meiosis 1 and meiosis 2. As a result of meiosis 1, 2 haploid cells are formed. One cell gets negligible cytoplasm, other cell retains complete cytoplasm. This is to avoid loss of cytoplasm. The larger cell is known as secondary oocyte. This is the stage where the egg is released from the ovary. The smaller cell is known as first polar body. It invariably degenerates. It never undergoes meiosis 2. The secondary oocyte undergoes meiosis 2. Again, nuclear division is proper, haploid and haploid, and one cell gets negligible, other cell gets maximum cytoplasm. So, this bigger cell which is formed is the egg or ootid. The other smaller cell which is formed is known as the second polar body. These polar bodies are also known as polocytes. Now, when this starts fetal stage and when it ends at fertilization, there is a long gap where this process remains suspended, and that is why it is known as a discontinuous process. When we saw spermatogenesis, in spermatogenesis it starts at puberty and then continues. There is no pause or no long suspension of any process. Whereas in case of oogenesis, there is a long phase where the maturation is arrested, and that is why it is known as a discontinuous process. So, when we wrote in spermatogenesis that it is continuous process, we said we will discuss about it. So, oogenesis is discontinuous, spermatogenesis is not, it is a continuous process. Oogenesis starts in the fetal life, gets completed after puberty, that too if fertilization takes place. Secondary oocyte is released from the ovary, it is going to come into the fallopian tube. If fertilization takes place, then meiosis 2. So, here let us write down this, that meiosis 2 will take place only if fertilization takes place. So, meiosis 2 only if fertilization takes place. As soon as the sperm enters, meiosis 2 will start. So, this is important for us to know. And another thing which is important is that after a short growth phase, it enters into maturation. So, here we can write one more thing, that the process gets arrested in prophase 1 of meiosis 1, and it restarts at puberty, and that is why we call it a discontinuous process. All the eggs which are produced, they are identical, because the sex chromosome which is present is X. So, only X containing, because females have only 2 X's. So, the egg will have, on anyways only one egg is going to be produced at a time. So, all eggs are identical, they are X containing eggs, whereas we talked of sperm, there are 2 types of sperms, X containing and Y containing sperms. Now, let us also see the structure of the fully formed egg or this one we have seen when we made the Graafian follicle. Now let us talk about the structure of the human egg.